Hello, I'm Jenny from AnnJi Pharmaceuticals, and it is an honor to be speaking to you. Thank you, IMARC, for the kind invitation. Today, I'm going to share with you the insights of our company and our program in the development of a treatment for SBMA. Our company focuses on therapeutics for neglected diseases, and we are developing novel drugs, in particularly for neurological and fibrotic diseases. Today, I will be only focusing on an NRF2 activator named AJ201, which is being developed for the treatment of SBMA. Our management team brings together decades of global expertise across the entire drug development life cycle. This is led by our CEO, who has over 40 years of experience and has successful drug development of more than 20 new drugs. To begin, I'd like to walk through the causes of SBMA. It begins with a mutation in the androgen receptor gene on X chromosome. Normally, this gene provides the instructions to make healthy AR proteins. However, in people with SBMA, this gene has extra repeats called CAG repeats and will cause the body to make unusual AR proteins that are longer than they should be and easier to fold incorrectly. These misfolded AR proteins tend to clump together in the nerves and muscle cells, and along with other factors like oxidative stress and inflammation, causes nerve damage and muscle atrophy, leading to muscle weakness. Our study drug AJ201 has shown some promising potential to help the body address this process. AJ201 can activate cellular cleanup systems to clear away the clump AR proteins. AJ201 can also activate the body's defenses against stress and inflammation, helping to protect both nerves and muscles. These mechanisms were found through preclinical studies, and in the next slide, I will be showing some of the results. Shown on the left side is a SBMA mouse model. The results show that AJ201 reduces clumped AR in mouse muscle. The bands over here are the AR protein clumps. We can see here that AR protein clumps are reduced in the mouse that was treated with AJ201. On the right side of this slide is a study that measures physical strength and coordination of SBMA mouse. In this study, we compared two groups of mice over several weeks. The gray line represents the group that did not receive the drug, and orange line represents the group treated with AJ201 daily from 10 to 20 weeks. You can see that starting around week 12, the mice without treatment began to lose muscle strength and had more difficulty with the physical task. However, the mice treated with AJ201, the orange line, showed a significant difference. The drug helped slow down the loss of muscle strength, allowing them to maintain their physical function for much longer. These results gave us the confidence to move forward into human studies. With that in mind, I would like to show you some results from our Phase 2 study in participants with SBMA. In the Phase 2 study, our absolute priority was patient safety. I'm pleased to share that the drug was well tolerated, with most side effects being mild and manageable. We also looked at how the drug works in the body. We saw a reduction in the toxic AR proteins that cause the disease, and we observed a positive shift in muscle-related laboratory markers like creatine kinase and myoglobin. Finally, we explored physical function. While this is a small early-stage study, we saw encouraging trends in walking distance and daily function. These results provide a strong foundation for our next steps in clinical development. Our next step is the Phase 3 pivotal study. We're aiming for this to be the final stage of testing required to bring this treatment to the community. The study is called ROMA, R-O-M-A, and is a 2-year study. Participants will be randomized to receive AJ201 or placebo in the first year, and all participants will receive AJ201 in the second year. Our main goal is to measure how AJ201 helps with the daily function, overall mobility, and quality of life. We are currently in active discussions with regulatory agencies in the US, Europe, and Japan to finalize the study details. We know how much the community is anticipating this, and we're working with the utmost urgency. Thank you for your time, and if you have any questions, please contact my colleague Andy. I look forward to sharing more updates soon.